We begin our journey in the microscopic world of prokaryotes. These single-celled organisms, including bacteria and archaea, are the smallest and most ancient life forms on Earth. Don't be fooled by their size. Inside these tiny cells lies a universe of genetic information encoded in their DNA. This DNA contains the blueprints for everything the cell needs to survive and thrive. However, managing this vast library of genetic information is no easy task. Imagine trying to find a single recipe in a cookbook the size of a skyscraper. Prokaryotes have evolved ingenious mechanisms to control which genes are active at any given time. This is crucial for their survival. They need to conserve energy and resources by only producing the proteins they need. This is where operons come into play. Operons are like genetic switches, turning genes on or off in response to the cell's environment. They allow prokaryotes to adapt quickly to changing conditions, ensuring their continued existence in an ever-changing world. Before we delve into the specifics of operons, let's familiarize ourselves with some key genetic components. Think of these as the tools that govern gene expression. Promoter. Imagine a construction site. The promoter is like the start here sign for the building crew, RNA polymerase. It tells them where to begin constructing a specific protein. Operator. Now picture a foreman on the construction site. The operator is like the foreman, who can give the go or stop signal to the building crew, controlling whether or not a protein is made. Regulatory gene. This gene is like the architect who drew the blueprints for the building. It produces a protein, repressor or activator that can bind to the operator, influencing whether the building crew gets to work. Structural gene. These are the actual blueprints for the building itself. They contain the instructions for making the protein that the cell needs. With these key players in mind, we can now explore how they work together in the fascinating world of operons. The Lac operon, found in E. coli bacteria, is a classic example of gene regulation in action. It controls the production of enzymes needed to break down lactose, a type of sugar. Think of it as a switch that turns on when lactose is present and off when it's absent. Here's how it works. No lactose. When lactose is absent, a repressor protein binds to the operator region of the Lac operon. This blocks RNA polymerase from attaching to the promoter and transcribing the genes needed for lactose digestion. The operon is off, lactose present. When lactose is available, it acts like a clever saboteur. It binds to the repressor protein, changing its shape and preventing it from attaching to the operator. Now, our RNA polymerase can bind to the promoter and transcribe the genes, producing the enzymes needed to break down lactose. The operon is on. The Lac operon ensures that E. coli only expends energy producing lactose-digesting enzymes when lactose is actually present. A remarkable example of cellular efficiency. Now, let's turn our attention to the Trp operon, another elegant example of prokaryotic gene regulation. This operon controls the production of tryptophan, an essential amino acid that bacteria need to build proteins. Unlike the Lac operon, which is induced, turned on by the presence of lactose, the Trp operon is repressed, turned off by the presence of tryptophan. It's like a factory that shuts down when it has enough product in stock. Here's how it works. Low tryptophan. When tryptophan levels are low, the repressor protein is inactive and cannot bind to the operator. This allows RNA polymerase to bind to the promoter and transcribe the genes needed for tryptophan synthesis. The operon is on. High tryptophan. When tryptophan is abundant, it acts as a co-repressor. It binds to the repressor protein, changing its shape and allowing it to bind to the operator. This blocks RNA polymerase from transcribing the genes, halting tryptophan production. The operon is off. The Trp operon ensures that the cell only produces tryptophan when it's needed, preventing the wasteful production of this essential amino acid. The Lac and Trp operons, while controlling different metabolic pathways, both exemplify the brilliance of prokaryotic gene regulation. They highlight how a single regulatory mechanism can be adapted to respond to different environmental cues. The key difference lies in their response to their respective molecules. The Lac operon is induced by lactose, meaning it turns on in its presence. On the other hand, the TRP operon is repressed by tryptophan, so it turns off in its presence. Think of it this way. The Lac operon is like a light switch that's turned on when it senses darkness, or rather, the absence of lactose. Meanwhile, the TRP operon is like a thermostat that's switched off when it senses the room is warm enough, or in this case, when there's enough tryptophan. Both operons demonstrate the remarkable ability of prokaryotes to fine-tune their gene expression in response to their surroundings. Let's try to understand these complex genetic processes with some everyday analogies. Imagine you have a recipe for a delicious lactose-laden dessert. You only want to make it if you have lactose in your pantry. The Lac operon is like your recipe book. It remains closed or genes off unless lactose is present. When you have lactose, it's like opening the recipe book and following the instructions to make your dessert or genes on. Now, imagine a toy factory that produces tryptophan toys. The factory has a sensor that detects the number of tryptophan toys in stock. When the stock is low, the factory runs at full speed, meaning genes are on. As the stockpiles of tryptophan toys increase, the sensor slows down production until the factory eventually stops, or genes are off when there are enough toys. These simple analogies illustrate the elegant logic behind operon function, turning genes on or off based on the availability of specific molecules. Operons are not just a fascinating quirk of prokaryotic biology, they have real-world applications that are shaping the future of medicine, industry, and biotechnology. Understanding how bacteria regulate gene expression, particularly through operons, is crucial for tackling the growing threat of antibiotic resistance. By targeting the operons that control the expression of antibiotic resistance genes, we can potentially develop new strategies to combat this global health challenge. Researchers are exploring ways to engineer bacteria to produce biofuels such as ethanol and butanol from renewable resources. By manipulating operons involved in metabolic pathways, we can enhance the production of these valuable biofuels, paving the way for a more sustainable future. Operons can be engineered to act as biosensors, detecting specific molecules in the environment. For example, modified Lac operons can be used to detect pollutants in water or heavy metals in soil, providing valuable tools for environmental monitoring. These are just a few examples of how our understanding of operons is being harnessed to address some of the most pressing challenges facing humanity. While operons are primarily found in prokaryotes, the principles of gene regulation they embody are universal. Eukaryotic organisms, including plants and animals, also possess intricate mechanisms to control gene expression, albeit with some key differences. Instead of operons, eukaryotes rely on a more complex system involving multiple regulatory elements and transcription factors that interact with DNA to control gene expression. Despite these differences, the fundamental principles remain the same. Cells must regulate which genes are active at any given time to ensure their survival and carry out their specific functions. As we delve deeper into the molecular world, our understanding of operons and gene regulation continues to expand. New discoveries are revealing the intricate details of these genetic switches and their role in shaping life as we know it. The future holds immense potential for harnessing the power of operons. By manipulating these genetic circuits, we can engineer organisms with novel capabilities, develop new therapies for diseases, and address some of the world's most pressing challenges. Operons, once a mystery hidden within the microscopic world of bacteria, are now recognized as fundamental components of life's genetic blueprint. As we continue to unlock their secrets, we open up a world of possibilities for understanding, manipulating, and even mimicking nature's ingenious designs.